Did you know that hearing loss in midlife is actually the single greatest modifiable risk factor for Alzheimer's disease?
It's a staggering statistic, really.
Right. And for decades, scientists have just been completely puzzled by this. I mean, we've known the numbers are high, but the actual mechanism behind it has been a total mystery.
Yeah,
Is
exactly.
it just the social isolation that comes with hearing loss that causes the cognitive decline? Or is there some indirect correlation we're just totally missing?
Well, today's deep dive is going to look at a groundbreaking new paper from Scientific Reports. And the researchers are suggesting something radically different,
Yeah, they believe our auditory system might actually provide this early warning window directly into the brain's functional state.
which goes far beyond just a story about hearing loss.
Exactly. We have this massive stack of complex neurophysiological research to get through. And our mission for you in today's deep dive is to really unpack exactly how scientists are using sound waves and these brand new mathematical models to spot Alzheimer's risk years before traditional symptoms ever appear.
Because we're essentially looking at how the brain's core circuitry fundamentally processes information.
Yeah.
The auditory system just happens to be uniquely positioned to show us exactly when that circuitry starts to misfire on a microscopic level.
Long before a patient ever complains about memory loss.
Right.
Okay, let's unpack this. Because to understand how we can literally hear Alzheimer's risk, we first have to understand how we actually measure the brain's response to sound.
And crucially, we need to understand why the old way of measuring it was systematically throwing out the most important clues.
Yeah, let's talk about the diagnostic tool itself first, the auditory brainstem response, or ABR.
Right. So if you picture like a rapid, non-invasive EEG that's specifically dedicated to your auditory pathway, you kind of have the right idea. A clinician places some minimally invasive electrodes on the scalp, plays a series of sounds into the ear, usually rapid clicks or specific pure tones, and then records the electrical neural transmission as it travels from the inner ear, up the brainstem, and into the brain.
And when you look at that recording, you see this series of peaks and valleys, right?
Right.
They're labeled wave 1, wave 2, wave 3, and so on, which represent different relay stations in the brain. But here is where the mathematical approach went completely sideways for decades.
It really did.
The traditional method for analyzing an ABR is to play that sound, say, 500 times. Then you average all 500 of those brainwave responses together to get one clean, standard, smoothed-out curve.
And we have to look at the historical context to understand why they did this in the first place. Back in the 1970s and 80s when these protocols were developed, computers simply didn't have the processing power
Right.
or the memory to store and analyze 500 individual microsecond-level waveforms. The hardware just couldn't handle it.
So they just deleted the raw trials.
Yeah, they had to compute the average on the fly and delete the raw data just to get a manageable file size. Mathematically, when you do that, you're actively deciding that any tiny trial-by-trial fluctuation in the brain's response is just random background noise.
You erase the variations to get a pretty picture,
Exactly.
but it turns out that noise was the actual signal.
Yes.
Traditional ABR is, it's like judging a city's climate by looking at its average yearly temperature. It looks perfectly stable on paper. But this new method is like studying the daily erratic wild temperature swings to predict a major climate shift.
What's fascinating here is that this empirical biological noise isn't actually random at all.
Okay, how so?
Well, the researchers in this paper discarded the averaging method entirely, and they started looking at what they call micromovement spikes.
Micromovement spikes.
Yeah, they looked at the tiny individualized fluctuations away from the local mean on every single trial. And they standardized these micropeaks to account for anatomical differences between subjects.
Like head size or skull thickness.
Yeah, right. So they could compare the actual neural performance apples to apples.
And when they mapped out these thousands of micromovement spikes, they found they perfectly fit a very specific mathematical model, a continuous gamma family of probability distributions.
Yes.
And I'm going to need you to break that down for us because continuous gamma family sounds like something from an advanced physics textbook.
I know, it sounds incredibly dense.
Mm-hmm.
But instead of assuming how the data should look based on a theoretical average, they let the raw, messy biological data speak for itself. They mapped this gamma distribution on a parameter plane with two axes.
Okay, an X and a Y axis.
Exactly. The x-axis is the gamma shape, which tells you about the type of distribution curve the brainwaves are forming. And the y-axis is the gamma scale, which essentially measures the dispersion or the noise-to-signal ratio of the firing neurons.
Can you give me a visual for that shape and scale? Like, how should we actually picture this?
Think of the scale on the y-axis like the volume of a crowd in a busy restaurant. It measures how loud and chaotic the background noise is. Then think of the shape on the x-axis like the actual rhythm of their conversation. If everyone is talking smoothly and taking turns, that creates a healthy shape.
Right.
But if they're suddenly shouting, interrupting each other, and pausing erratically, the mathematical shape warps. That warped shape is essentially the neurodegenerative stutter.
Oh, wow. So by tracking the volume of the noise and the rhythm of the firing, they created a highly sensitive, individualized mathematical fingerprint for every single subject.
They did. They tapped into the vital signal of synaptic health that older computers just, well, forced everyone to average out.
But having a mathematical fingerprint is really only half the battle. To prove this gamma shape actually means Alzheimer's, you have to test it on a brain you know is destined to develop the disease.
Right. And to do this, the researchers used a preclinical rat model, specifically rats with the homozygous Swedish risk variant knocked into their genes. They compared these to control rats with a healthy, humanized gene variant. We'll call the at-risk group the APS rats, and the healthy control group the APH rats.
I have to pause you there, though, because whenever we talk about rat models and Alzheimer's, there's always a massive caveat, right? Rats aren't humans, and fixing a rat brain doesn't mean we can fix a human brain.
Absolutely.
Plus, homozygous Swedish risk variant knocked into their genes is incredibly dense jargon. How confident are researchers that these specific rats actually mirror human brain chemistry?
It is a really vital distinction to make. When geneticists say a trait is knocked in, they mean they have used gene editing tools to insert the exact, specific human DNA mutation that causes early-onset familial Alzheimer's directly into the rat's genome.
So they aren't just normal rats with age-related memory issues.
No, not at all. They are biologically programmed to develop the exact same amyloid beta plaques that humans with this genetic variant develop.
And what's crucial for you, the listener, to know about these specific rats during the test is that they are in a prodromal state, which is a medical term meaning they do not yet have the classic Alzheimer's plaques or tangles actually forming in their brains.
Exactly. This mirrors human midlife, like your 40s or 50s, which is exactly when hearing loss first emerges as a dementia risk factor. The researchers even tested their hearing with standard click tests, and the rats had totally normal hearing thresholds. They were not deaf.
Yet, when the researchers ran the single trial ABRs and plotted those thousands of micromovement spikes on the gamma parameter plane, the results were just staggering.
They were. The APFS, the ones with the invisible Alzheimer's risk, separated completely from the healthy APH rats on the graph.
And the researchers noted 95% confidence intervals showing zero overlap. We need to translate that into visceral imagery. Zero overlap means if you plot this on a visual graph, the healthy brains are all huddled together tightly in one corner, and the at-risk brains are isolated in a completely different corner.
There is no gray area.
And here's where it gets really interesting. They found the disease signature before the disease even structurally formed. I mean, they saw this complete separation in aged rats. But remarkably, they also saw it in young rats that were just 5 months old.
Yeah. And to understand the biology of why this is happening so early, we have to zoom all the way down to the synapse, the gap where two neurons communicate.
Okay.
The gamma parameters are picking up on microscopic functional imbalances in synaptic transmission. Specifically, they're detecting an imbalance between glutamate, which is a neurotransmitter that excites neurons and tells them to fire, and GABA, a neurotransmitter which calms them down and resets them.
It's like the timing belt in a car engine starting to fray.
That's a great way to look at it.
The engine hasn't blown up yet. The car still drives down the highway just fine. But the microstutters, the tiny timing misalignments between the pistons, are there if you have a sensitive enough sensor to measure them.
And the auditory system is the ultimate sensor for this. Processing sound requires the fastest, most temporally precise neural activity in the entire mammalian brain.
Wow.
It operates on a microsecond scale. It is incredibly demanding on the brain's energy reserves. If there is a synaptic stutter, a slight delay in that glutamate and GABA reset anywhere in the central nervous system, the auditory brainstem response will catch it first because it simply cannot afford to miss a microsecond.
So we have this mathematical signature. It's so reliable that we can plot it cleanly on a graph, separating the healthy from the at-risk with zero overlap. Because it's highly quantifiable, this gamma data becomes the perfect fuel for the future of automated medical technology.
Exactly. We're talking about feeding this into machine learning algorithms.
Right.
When the researchers took this data and plotted it on a log-log scatter plane, they found a very tight linear relationship between the two parameters. As the scale parameter, the dispersion we talked about earlier, decreases, the shape parameter increases. They move in lockstep.
Wait, log scatter plane and moving in lockstep, you're losing me a bit in the statistical jargon there. Let's break down how the AI actually uses this.
Right, sorry. A log-log scatter plane is simply a mathematical trick scientists use to squash giant ranges of numbers onto a single graph so they can actually see the relationship between them.
Oh, okay.
When they did this with the brainwave data, all the dots formed a clean, straight line. Because of this tight linear fit, knowing one parameter allows an algorithm to instantly infer the other. It reduces an incredibly complex, chaotic web of brainwave data down to its inherent noise-to-signal ratio.
And that is a goldmine for artificial neural networks. But wait, why is this gamma data better for AI than the mountains of traditional medical data we already have? Hospitals have, millions of standard EEGs and MRI brain scans sitting on file.
Well, a machine learning model is really only as intelligent as the reality of its training data. Currently, a lot of predictive models rely on assumed theoretical data distributions. They use those smoothed-out averages we talked about earlier. If you train an AI on smoothed-out averages, it only learns to look for massive, obvious craters, like severe cognitive decline or physical brain atrophy. It completely misses the microcracks, which leads to massive amounts of false negatives.
Because it's looking for a broken bone when it should be looking for a stress fracture.
Exactly the issue. But by feeding the AI these actual stochastic biological nuances,
Which just means the messy, unpredictable realities of a living, breathing cell rather than a mathematically perfect fake curve.
yes. By feeding the AI the messy reality of the living synapse, the AI learns what real early-stage neurodegeneration actually looks like on a cellular level. It learns the exact mathematical boundary of a healthy brain.
Wow.
If a patient's data deviates from that specific line on the log plane, the AI instantly flags it as abnormal. It can automate the classification of disease states and track tiny progressions over time that a human doctor would just never spot with the naked eye.
That is wild. We are literally teaching algorithms to read the microscopic stutters of our brain cells.
Yeah.
But here is the part of the paper that genuinely made me sit up in my chair.
Yeah.
We know the gamma signature can perfectly identify Alzheimer's risk, and we know how to train AI to spot it. But what if we try to actively intervene? What if we try to change the brain's mathematical signature?
This is where the researchers introduced an active behavioral intervention. They put the rats through a two-tone discrimination task.
Yeah.
They played a 5.0 kilohertz tone, and if the rat pressed a bar, it got a water reward.
Okay.
Then they played an 11.5 kilohertz tone, and if the rat pressed the bar, it got a flashing light and a timeout penalty.
And we shouldn't think of this like a normal hearing test, right? Because 5.0 and 11.5 kilohertz are very far apart acoustically. Any rat can hear the difference.
Yeah.
They essentially put these rats through a grueling memory game. Hear a high pitch, press a lever, get a reward, hear a low pitch, do it, and get a flashing light and a timeout. It was forcing their brains to deeply focus, categorize the sound, and assign meaning to the frequency.
It was a test of auditory-cognitive memory. It requires the auditory cortex to deeply engage with the incoming sound waves and build a predictive model of the world. And they trained the rats for just 2 to 3 weeks.
And the revelation here is massive. After this short, intense bout of auditory training, they ran the ABR brainwave test again. The gamma signature of the at-risk app's rats literally shifted on the graph.
It did.
It moved completely out of the disease risk isolated corner and physically overlapped with the healthy control rats.
The cognitive training functionally rewired their neurophysiological signature. And what's more impressive is the longevity of this effect. This protective, healthy shift in their brainwaves lasted for up to 9 months.
Which is huge.
In a rat's lifespan, which is only about 2 to 3 years total, 9 months is a remarkably long period of resilience.
They also noticed a structural change in the brainwaves to back this up, didn't they?
Yes. The researchers found that this behavioral improvement correlated with a massive increased amplitude in what is called the second peak wave, or PW2, of the auditory brainstem response. And that amplitude increase scaled directly with how well the rats actually remember the tone task.
So the better they learned the auditory task, the stronger that specific neural pathway became.
Exactly. The physical voltage of the brainwave increased. fundamentally overriding the genetic risk signature in the data.
Okay, so what does this all mean? Did they cure the Alzheimer's mutation by making the rats play a memory game with beeps?
Well, if we connect this to the bigger picture, it is crucial to state that this is not a cure for the neurodegeneration itself.
Right.
The rats still have the Swedish mutation embedded in their DNA. The amyloid beta proteins will eventually still form. What this proves is that the auditory system has incredible experience-dependent neuroplasticity.
Meaning the brain changes its physical structure based on what it experiences?
Yes. By forcing the brain to actively listen, learn, and categorize sound, it builds functional resilience in the brain's circuitry. It essentially fortifies the synaptic pathways, allowing the brain to maintain healthy signaling and keep the glutamate and GABA balanced, despite the genetic risk factors operating in the background.
It's building neurological scaffolding. Which brings us to the ultimate application for you, the listener. We've seen these incredible graph-shifting results in rats, exploring how synapses fire and rewire. But how does this translate to human medicine? Where does this diagnostic tool go next?
The translational potential for human diagnostics is massive. Think back to the benefits of the auditory brainstem response. It is totally non-invasive, it relies on cheap, widely available electrode equipment, and it's rapidly acquired.
Yeah.
But critically, the ABR is a pre-attentive signal.
Pre-attentive is a huge point. It means the patient doesn't have to actively pay attention or perform a complex, frustrating memory task to get the reading.
Exactly the opposite of current cognitive tests. Testing someone who is already experiencing cognitive decline with a grueling behavioral exam causes intense stress, anxiety, and often yields unreliable results.
Totally.
The ABR is entirely passive. You just place the electrodes on their scalp while they sit there quietly, and the brainstem automatically processes the sound waves.
Imagine going in for your routine physical in your late 40s or 50s. Alongside checking your blood pressure and your cholesterol, the nurse puts some sticker electrodes on your head, plays a few minutes of rapid clicks into your ears, and that normal hearing test becomes an early brain health screening.
Yeah.
They use the gamma mathematical model to see if your synapses are micro-stuttering.
This raises an important question about how we fundamentally handle dementia in modern medicine. Currently, our entire system waits for a patient to complain about memory loss or behavioral changes. By the time that happens, the structural damage to the brain is severe and irreversible.
Too late.
Right. This ABR tool flips that entire paradigm. It allows for proactive midlife prevention. With this rich mathematical parameter space, we could eventually achieve true precision medicine, stratifying different types of dementia, identifying specific genetic mutations long before any clinical hallmarks appear.
Wow.
This allows doctors to tailor individualized interventions like auditory training or early pharmacological treatments right when they can do the most good.
So to recap this incredible journey, we started with a decades-old statistical flaw, discovering how older technology forced us to throw out the most vital data by averaging brainwaves.
Oh.
By designing new math to dive into the messy biological noise, researchers discovered the hidden gamma signal that perfectly flags Alzheimer's risk by measuring synaptic stutter. And most hopefully, we learned that intense cognitive auditory training can actually functionally rewire that risk signature, building physical resilience in the brain's circuitry.
It fundamentally changes how we view the relationship between the ear and the brain. The auditory pathway isn't just a passive microphone collecting sound. It is a direct, measurable window into the core health of our central neural networks.
Which leaves us with a final unaddressed thought to ponder. This study proved that intense auditory cognitive training forcing the rats to deeply focus and decipher meaningful sounds built lasting resilience in their brain's wiring. If that is true, could our own daily listening habits be acting as a daily invisible brain gym? Think about the mental effort of struggling to learn a new language, deeply analyzing the time signatures in a complex piece of jazz, or actively trying to pick out the hidden bass line in a crowded song. Could that active, focused listening be secretly building our neurological armor against cognitive decline?
It's a fascinating thought.
We might not have that perfect, clean, engineering-grade X-ray for Alzheimer's just yet, but we are finally learning how to listen for the tiny fractures in the noise. Thank you for joining us on this deep dive. Keep listening closely, and we'll see you next time.